And with that, I would love to introduce my friend, collaborator, amazing colleague, amazing mathematician, great human being. I wish we had the presentation also up. So you have a stand-up at 12 minutes.
Okay.
And you hear that.
You just shut at me, right?
Yep.
Cool. Okay. Thank you. I'm not really an amazing human. I'm just okay. I just want to echo Luciana's comments before. Many, including obviously the love for ABMs, which I share, but also that coming from Glasgow, coming here is a great. So just a lot of us woke up this morning in Tampa, covered by snow, and after a 1 hour plus driving, we're here. So it's incredible to be here in St. Pete. Wake up in the morning, see the Gulf of Mexico. Is this, can I say that? So it's great to be here, and I'm a local, just basically what I wanted to say. I don't sound like one. So I'm one of the guys in Cancer Evo, and you can see a bunch of the people I work with in the picture on top, and some of us are actually here presenting. Tatiana is presenting the next session, so I'm looking forward to her presentation. And like Maxi said before, collaborations are super important in math modeling, and having deep, strong collaborations are key. And the presentations I'm going to sort of discuss today involve two key collaborators, Andriy Marusik and Connor Lynch. So I'm definitely on the same page when it comes to the importance of really not just getting data from collaborators. I think data is important and is crucial to us and to our models, but the insights and the grounding you get by actually sort of trying to convince an experimentalist that what you're doing makes any sense is a neglected sometimes part of the collaboration. So we do a bunch of things in the group, and what I like to group them by is by our focus on cancer evolution. We believe cancer evolution is key. We don't believe that genes are so important as Mehdi said before. If you're in trouble with saying these things in public and recorded, I am also in trouble now. So, you know, but my career is not as important as yours. But nonetheless, I said it, genes are overstated when it comes to their importance in cancer evolution. It's the easiest thing to record and track over time if you are an experimental biologist, but doesn't make them super important. They are key and they do have an importance, but in my mind, the thing that usually people don't look at when it comes to what drives cancer is the ecosystem. Luckily for you guys, you had Mehdi talking about the breast ecosystem before, and I'm going to be talking about other ecosystems. I had this beautiful picture someone else made some time ago about the bone ecosystem, and I use it all the time because it's a great representation that when it comes to cancers in an ecosystem, looking at the cancer cells themselves is not the only thing. So there's a bunch of macroenvironmental factors, biophysical factors, treatment, cell types that actually drive what's going to happen to this cancer. So we have a bunch of questions we're trying to address, and I'm dropping it here because I know that we have another 3.5 days in this meeting, and some of you might actually find some of these things interesting, and you might want to talk to me about them. There's going to be lots of coffee breaks. Some of you might go to the beach. I'll still have my coffee. So we believe that heterogeneity is important. In case someone from the government is looking, I hate diversity. I think diversity is the strength of cancer. It makes cancer really able to withstand lots of different challenges. So we really want to make sure we understand how heterogeneity happens and how to fight it. We want to fight diversity in cancer. I still have my job, right? So with Mehdi, actually, we're looking at these things, and I think this is something that if you find time in the coffee break, just talk to us. But we also care about aging. It's important because a lot of the studies we do on cancer comes from mouse models that are adolescent mouse models. We think we can do better than that, and we think that math models are the way to actually do that. We want to study adaptive therapies. This is something that Maxi talked about before. We also are interested. Tumor-stromal interactions are super important. We want to use AVMs to model this. The mutated phenotype is something that I might not be discussing today, but Gosiawik, which is a former PhD student in the group who is now working with Heiko, will be here this week. So she and I can actually talk about this. And also niche construction metastasis, which is something we also are very interested in, and which I believe is Tatiana's topic in an hour. So let's start with the first project. I'll try to go as quickly as I can. I don't know how much time I've got left, but hopefully some, because I haven't even started. Multiple myeloma, important disease. And this is a project I started a while ago. Anna Miller-Gaffney, who used to be an integral part of the group, and now she's making the big bucks in a company called Certara, consulting for pharma companies, and now taking over by Matt Foy, with a PhD student who's definitely not making the big bucks, and who's working on continuing that work in the world of immunotherapies. And this is in collaboration with Elaine's lab, who's also at Moffitt. So multiple myeloma, in case you just want to have like a very quick one-on-one, it's just basically this hem-onc disease that, is quite common. It's not the top hem-onc disease, but it's actually something that happens to a lot of Americans every year. It's treatable but not curable, which basically means we have treatments and eventually these treatments fail and tumor relapses. So we're trying to understand this better. And the way we did it is through an agent-based model, because that's the kind of thing we do in our group. And you see this picture on the left. It's just basically like a very tiny window inside the bone marrow of a patient that has multiple myeloma. So basically there is trabecular bone, which is just bone, lots of calcium deposits that you can use in your body, and you require calcium in a hurry, and something that gives structure to the bone. But basically this bone actually is a very dynamic organ. So we start with this, just bone. There's a bunch of cells in the bone that are doing their things. They do the things until cancer happens. And we wanted to model this. There are mesenchymal stem cells, there are osteoblasts. They usually go and remodel bone all the time. So bone gets remodeled every 5 years. And we wanted to capture this first and then figure out what does myeloma do to that. So on the left, you have just normal bone remodeling when there's no cancer. On the right, you see myeloma. And you can see how on the left, we managed to capture what we feel are the main dynamics of normal bone remodeling. So cells come in, osteoclasts come in, destroy bone that needs to be reconstructed. Osteoblasts come in and start constructing bone. This is something that emerges naturally. This is one of the beautiful things about agent-based models. You define a few rules, and the emergent behavior is really what you're looking after. So what happens on the left is what we know happens in bone remodeling. What we see on the right is what happens when you inject a bunch of tumor cells and let them grow. And obviously, if you have a bunch of tumor cells with the right phenotype and nothing to stop them, this is what happens. Our question is, okay, we know there's treatment, we know that treatment fails, so what is the role of the microenvironment in that? What's treatment? So there's a bunch of treatments for multiple myeloma, there's a line of treatments, and the standard one for a lot of time, for a lot of people, was something called proteasome inhibitors. How we model them is not mechanistic, but it's basically something that impacts tumor cells that are sensitive. And if you're sensitive, you die by it, and if you're resistant, you don't. But the microenvironment can play a role, because we know the microenvironment, so tumor cells that happen to be in contact with the bone as it's being remodeled, in contact with certain cells like MSCs, baited by factors like TGF-beta, are more likely to resist treatment. So we model this because I'm not going to show you all the data we got. There's a whole paper. I'm happy to share the slides when I had more time, but we had a lot of experimental data to support every one of the model's assumptions. So on the left, tumor cells, no treatment. In the middle, we have now environmental mediated drug resistance. So there are niches in the environment that support the survival of tumor cells that will be sensitive to treatment and are preserved from dying by the environment. On the right, we just decided to figure out what would happen if there was actually no microenvironment protecting the tumor. This is one of the beautiful things about the agent-based models, or any mathematical model, which is you can play a bunch of scenarios and see what would the outcome be. So loss of growth in one. The middle, you can see tumor cells start dying, but in the middle, they still remain behind. On the right, tumor cells die, but not all of them. You can see the black cells coming up. Those are Führer-resistant cells. They are completely insensitive to treatment. So on the one on the left, tumor grows as expected. The one in the middle and the one on the right, if you wait long enough, they actually look quite similar in terms of the outputs. In both of them, if you are just a pathologist and you don't have this beautiful green-black coloring to figure out what kind of phenotype of myeloma cell you got, they look kind of the same. But there's a big difference. The one in the middle is the kind of resistance that emerges when you have environmental mediated drug resistance. The environment helps protect tumor cells. It creates this room for minimal residual disease to maintain. On the right, we actually have just a lucky case where the tumor had one lucky phenotype that was resistant. Mutations occur every time there's a division, so you have this lucky mutation that was present before treatment and was selected very strongly because of the treatment. So what does this mean from the point of view of the clinic? Both of them look like the tumor came back, so what's the difference? Who cares about EMDR? Well, evolutionarily, there are very different outcomes. If you have EMDR, you are more likely to actually find that lucky clone just because you have preserved a lot of sensitive cells and you allow them to basically keep proliferating and dying until they find the right phenotype. On the case where you don't have EMDR, then you have to be very lucky. On top of that, you see these Muller plots. These Muller plots allow us to follow the population over time and see the different heterogeneity you see. So we tried different mutation rates. Just let's look at the bottom row. If you have no treatment, basically the tumor grows, and there's no reason for emergence of different clones to appear because they don't provide any particular evolutionary advantage. You see on the right, what happens is you apply treatment, and you see here exactly is where treatment is applied. And there was this lucky clone, and this lucky clone got selected, and this lucky clone is just going to repopulate the entire tumor. It grow up to the point in which you can't tell that you applied treatment in the first place. Here, though, you apply treatment, and the bottleneck is much thicker. There's a bunch of tumor cells that were sensitive and now are still there because they're protected by the environment. So the result is that you have a heterogeneous tumor cell population that is sensitive but protected, from which resistant clones will eventually emerge just because they can keep trying to proliferate and try different mechanisms to deal with the treatment. So not only you are more likely to get recurrence tumors in the case of EMDR, but also you're more likely to have a heterogeneous tumor coming from that, which from the treatment perspective is very different. If you're a doctor, you better treat this patient who has one driver that you can potentially find one target for versus this tumor for which there's no hope that you'd find one treatment that would target all these different populations. So this is where Anna left, and at some point, Matt came, and Matt is very interested in immunotherapy and T cells. And basically what you found is, well, CAR Ts are very popular these days, but BiTEs, or these T-cell engager therapies, are even more popular. The reason they're more popular is that they have a very similar efficacy while being much, much cheaper. And they basically still rely on engineering a T-cell population that targets a particular mutation. So that's what he did. He basically, I don't know how much time I got, and tried to go as quickly as I can. But this therapy still fails. As much as we like it to work, it fails. And there's a bunch of reasons why it might fail. So clones might actually lose the BCMA flag that T cells are looking for. It might actually sort of be T cell exhaustion of the microenvironment. And if you ask Nina, who is around here, she also says something about exhaustion of T cells. So there's a different set of reasons that might explain this. And mathematical models would allow us to basically explore the relative importance of these different mechanisms. So once again, we actually have lots of experimental and clinical data to support that different hypotheses, that Tregs may be predictive of responder status, that the antigen loss can contribute to DCA resistance, and this is important because we want to have a motivation to suggest whether tumor cells that lose the BCM flag might actually sort of have to pay a cost of resistance, which would allow us to introduce adaptive therapies, which Maxi said rely on the assumptions that the cost of resistance has to be meaningful enough for you to be able to modulate treatment in order to not select for resistance. So I'll just, once again, swim through this very quickly. I'm happy to talk about this during the coffee break, but there's a lot of data that has been used in order to create a version of this model where we have now CD8 cells, which are the cytotoxic, the cytokiller ones, and Tregs, which obviously are the ones that tumor cells can co-opt in order to sort of make sure that the tumors survive. So that's one of the mechanisms that allow the tumor cell to interact with the stroma to maintain survival over long periods of time. And we have data to support the initialization of the model, which allows us to make simulations and contrast them against experimental data. And the model now recapitulates certain dynamics in terms of CD8 populations, which are the killer T cells, and in terms of T regulatory cells. And we're going to be using this in order to create simulations that allow us, just to start with, figure out why certain patients respond and which patients don't respond. This is just a starting question. I think it's the easier one to address, but more importantly, once you understand this, I think it's much easier to figure out how to change the treatment so that you actually get every patient to respond. And it seems I might not have time to go over some of the other slides I had, which kind of I was afraid that was going to be the case, but yeah, I'm more than happy to go over the remaining projects I have if you have any time during the next 3 or 4 days. But yeah, I'd like to thank our collaborators, the people in the group, also people who have come to this conference and which I'm trying to maintain collaborations. Ronoble, Russ Rockney, which might come in a couple of hours. Mehdi, thank you very much. June, who's I know that just arrived, and Simon Seeger. And just as said I, we also have a PhD program. The deadline is 1st of December. So I know that you are not looking for a PhD, but you might know people who do. So thank you.
Thanks so much, David. Right on time. Perfect. We have time for 1 or 2 quick questions. Just to clarify, you say there's the genetic, and you don't believe it, and then you show the first model is like clonal evolution by mutation. How's that? Is it a phenotype that you talk?
So I think that what my first model shows is that the environment is what allows for the dynamics to be shaped in a particular way. If you have the right phenotype, you might actually be very strong and still go without caring about what the environment is. But what this model suggests is that whether the environment is supportive of tumor growth or not will definitely dictate whether the tumor is going to be going one particular trajectory or the other. That trajectory is super important from the point of view of how you treat the tumor. You and I want to make sure that we reduce heterogeneity, for instance. That's something important for us, right? So you have to be very careful when you apply treatment and how you apply treatment. You need to understand not only the tumor and just doing a biopsy of the tumor and sequentially finding the genes that drive that tumor might not be enough. You actually want to take a good look at the microenvironment as well and trying to understand how the microenvironment is going to respond to your treatment as well.
Perfect. Thank you. We're done?
Thanks, Mary.
I thought you promised 3.
No, he's understood. Anyway, thanks, David, again.
Thank you. Thank you so much.